Passive transportation is the transport across a concentration gradient that doesn't require any energy. Osmosis. Osmosis is the passive transportation of water across the concentration gradient. It helps to even out solute concentration without having to make solutes themselves. Water moves from a hypotonic environment to a hypertonic environment to try and create an isotonic environment. Hypertonic solution is a solution that has more solutes and less water. A hypotonic solution is a solution that has fewer solutes and more water. An isotonic solution is a solution with balanced solutes and water. This is the purpose of osmosis and diffusion. Osmotic pressure is when water moves into a cell by osmosis. Osmotic pressure may build up inside the cell. If a cell has a cell wall, the wall helps maintain the cell's water balance. Osmotic pressure is the way in which many plant cells obtain support. When a plant cell is in a hypotonic environment, the osmotic entry of water raises the turgor pressure exerted against the cell wall until the pressure prevents more water from coming into the cell. At this point, the plant cell is turgid. Diffusion. Diffusion is the movement from a higher concentration of solids to a lower concentration of solids in order to equalize the concentration. One type of diffusion is simple diffusion. This is the unassisted passage of small hydrophobic nonpolar molecules from a higher concentration to a lower concentration. The other type of diffusion is facilitated diffusion. Facilitated diffusion is passive transport that uses integral membrane proteins to help larger charged hydrophilic and polar molecules cross the concentration gradient. Transportation proteins are proteins that span the entire membrane and allow certain molecules or ions to diffuse across the membrane. There are three types. A channel protein, which is a type of transport protein, and it acts like a pore in the membrane that lets water molecules or small ions through quickly. Water channel proteins allow water to diffuse across the membrane at a very fast rate. Gated channel proteins is a transport protein that opens a gate, allowing a molecule to pass through the membrane. Gated channels have a binding site that is specific for a given molecule or ion. Lastly, there's carrier proteins. Carrier proteins are transport proteins that is specific for an ion, molecule, or group of substances. Carrier proteins carry the ion or molecule across the membrane by changing shape after the binding of the ion or molecule. Carrier proteins are involved in passive and active transport.
Active transportation is transport by pumping ions and molecules across concentrate gradient that requires chemical energy. Integral membrane proteins, called receptor proteins, recognize and bind to a specific target. When a receptor protein binds to a target, it signals the membrane to enclose the target and internalize it inside a special vesicle. Three types of protein that uses energy to transport against the concentration gradient are uniport. Uniport moves one ion or molecule in one direction. Antiport moves one ion or molecule in one direction while also moving another ion or molecule in the opposite direction. Or symport, which moves two ions or molecules in the same direction.
There's two types of active transportation. The first is endocytosis, which is the process of capturing a substrate or particle from the outside of the cell by engulfing it with the cell membrane. The second is exocytosis, in which vesicles fuse with the plasma membrane to release material out of the cell.
Endocytosis is the process of capturing a substance or particle from outside the cell by engulfing it with the cell membrane. Phagocytosis is when the plasma membrane dissolves materials and forms a phagocytic vesicle. Pinocytosis is when the plasma membrane folds inward to create a channel which allows dissolved materials to enter the cell. The liquid is encircled in a pinocytic vesicle when the channel closes. In the third type of endocytosis, the cell membrane also folds around a substrate to engulf and internalize it. In receptor-mediated endocytosis, this is a process that recognizes and targets a specific substrate through the help of integral membrane proteins on the cell membrane surface.
In exocytosis, the particles that cannot be digested by a lysosome are carried by a vesicle to the cell membrane. Because these vesicles are made out of the same phosphobilayer layer as the cell membrane, they can merge with the cell membrane and release their contents outside of the cell. The vesicle then becomes part of the cell membrane.